Good morning. My name is Jake Lesnick, and it is my pleasure to welcome you to today's seminar on development and applications of CRISPR Cas9 for genome editing. This online seminar, part of the BioConference Live Genetics and Genomics event, is sponsored by System Biosciences, SBI. Based in Mountain View, California, SBI is committed to developing innovative life science research tools to accelerate your discoveries. In April 2013, we launched our Cas9 Smart Nuclease system, becoming the first commercial source for CRISPR Cas9 reagents. Available worldwide, we have since expanded our portfolio of Cas9 products and related genome engineering reagents to now offer Cas9 mRNA, lenti-Cas9 plasmids and viruses, mutant Cas9 derivatives, gRNA libraries, and a suite of popular HR donor vectors. We provide detailed application notes and comprehensive technical support by phone to ensure our customers' success, and also offer Cas9 guide RNA design, cloning, and cell line engineering services. Let SBI be your guide to success with CRISPR Cas9 technologies. And now, to share a whole lot more about the development of CRISPR Cas9 tools and their potential to revolutionize research and medicine, it is my honor to introduce you to a true pioneer of CRISPR Cas9 technology, Dr. Feng Zhang. Dr. Zhang is a core member of the Broad Institute of MIT and Harvard and the W.M. Keck Career Development Professor of Biomedical Engineering at MIT. His lab works on developing and applying disruptive technologies, including optogenetics and genome engineering, both TAILS and CRISPR, to understand nervous system function and disease. He is a recipient of the National Science Foundation's Alan T. Waterman Award, Pearl UNC Prize in Neuroscience, the NIH Director's Pioneer Award, and awards from the Searle Scholars Program, McKnight, Keck, and Damon Runyon Foundations. Since his seminal 2013 science paper on mammalian genome engineering using CRISPR-Cas9 systems helped set off the explosion of research in this field, Dr. Zhang has published more than a dozen additional papers to continue driving CRISPR-Cas9 research forward, with his latest Nature paper published just 2 weeks ago. You simply could not ask for a more informed speaker to share his expertise on this exciting technology. So without any further ado, I welcome Dr. Feng Zhang to the virtual stage.
Good morning. Thank you for attending the webinar. And this morning, I want to tell you about some of the work that we have been doing to develop and also extend the applications of the CRISPR-Cas9 system for studying genomic functions in mammalian cells. So one of the main motivations for developing this technology was inspired by the rapid advances that we have made in human genome sequencing. So since the early 2000s, our ability to analyze genetic variations across large populations of individuals has really exponentially grown. And so over on the bottom of this slide, you can see a plot from the National Institutes of Health, and it reports the number of publications that describe genome-wide association study results. One thing to remember is that all of these genetic variations that the papers reported are based on correlation. And so the next challenge is to figure out which ones of these variations actually have a causal role in a biological process. So given this very rapid rise in the number of hypotheses that we generated, we need new methodologies to help us quickly follow up and test the functions of these mutations. So one way to test the function of a genetic variation is to directly put it into the genome. And so one way to do this is by using site-specific nucleases. Nucleases can introduce a double-strand break within a specific target locus, and this break is recognized by the cell as DNA damage. And that will activate one of two major pathways for repairing this DNA double-strand break. So one of the pathways is called non-homologous indjoining. This pathway does not require any DNA template, and it simply rejoins the two broken pieces of DNA back together. When this happens, the enzymes that are responsible also introduces a small scar. And this small scar can often lead to the formation of premature stop codon and allow you to knock out a gene function. Alternatively, the second pathway is based on homology-directed repair. This is very similar to the methods that we would use to generate a transgenic mouse knock-in by providing a repair template that carries the insertion sequence and also flanked by homology regions. We'll be able to introduce a new sequence. The difference between this and what many scientists have already been using now to make knock-in is that the double-strand break can significantly increase the efficiency of the editing. And so it can go from very low to something like 10 to the minus 6 to now in the single or even double-digit % range. So in order to realize these applications, we need to be able to introduce a double-strand break within a specific region of the genome. There have been multiple technologies developed by many different labs around the world, and so two of the prominent ones include zinc finger nuclease and also transcription activator-like effector nuclease, also called talons. Both of these nuclease technologies depend on protein DNA interaction. So the idea is that you can engineer the protein sequence to get it to recognize a new DNA sequence. Taking this DNA binding domain, we can fuse onto it a nuclease domain so that we can target an endonuclease activity to a specific genomic sequence. And so this is very powerful because we can now use these customized proteins to target virtually any genomic region we want. The challenge of using either zinc finger nuclease or talons is that protein sequences are difficult to manipulate, and so they require the engineering of novel proteins, which can take a decent amount of time and quite sophisticated molecular biology techniques. And so given the rapid rise in the number of genetic mutations that we want to study, it will be much nicer if we can have a simpler method to target genomic sequences. And so a few years ago, when I started my laboratory, we ran across a couple of papers that described a bacterial adaptive immune system called CRISPR-Cas. And it turns out that CRISPR-Cas is a nucleic acid nuclease system that uses short RNA guides to be able to target a double-strand break. So the work by Sylvain Moyneux and also Emmanuel Charpentier in Sweden, they started to pave the way for elucidating all the components that are necessary to engineer a CRISPR-Cas system. So in the past, Cas9 also was known as Cas5, CSN1, or CSX12. So what is the CRISPR-Cas system? So in nature, bacterial cells will constantly get invaded by phages or even conjugating plasmids. So to defend themselves against these infectious agents, the CRISPR-Cas system adapts small bits of nucleic acid information from previous infections and then remembers it, and those nucleic acids serve as an immune memory. So during subsequent infections, this short piece of nucleic acid information can get transcribed into RNA, and this RNA is used by a complex of enzymes to be able to recognize the invading DNA. And if there is recognition in the form of Watson-Crick base pairing, then the enzyme can get activated and it will cleave the invading nucleic acid. And because this is an adaptive system, so depending on different kinds of viruses that are infecting the cell, new forms of nucleic acid or RNA memory can get acquired, and this makes it easy to reprogram to target a new DNA sequence. And so when I initially started the lab, three very talented graduate students joined me, and we started to explore, can we harness the CRISPR-Cas9 system from two different kinds of bacterial organisms to be able to target a specific sequence within the human endogenous genome. And initially, we took 4 different components from the bacterial CRISPR system. This includes the Cas9 nuclease enzyme, and then we reprogrammed the CRISPR RNA to encode a piece of human DNA so that we can target the human genome. And then along with these two, we also introduced a bacterial RNA called tracer RNA, which hybridizes with the CRISPR RNA to load onto Cas9, and then also the bacterial RNAs, which is thought to be important for processing the CRISPR RNA into the mature form. And then by targeting the EMX1 locus, we were able to see whether or not we can achieve a targeted double-strand break within the human cell. And so when we introduce all four components, the Cas9 enzyme, the RNAs, the tracer RNA, and also the reprogrammed CRISPR RNA, we were able to detect double-strand breaks within the target human locus. And then at the same time, we found that if we don't provide the RNase molecule, we can still get cleavage. So that suggests that there may be compensatory RNases within the human cell that can also provide the maturation function. And so just by using Cas9, along with the two CRISPR RNAs, we can now edit the human genome. So while we were doing the development in human applications, a couple of other biochemical studies were published, and one of them is from Emmanuel Charpentier and Jennifer Doudna, and another one from Virginijus Siksnas in Lithuania. So one of the results from Dr. Charpentier's work is that you can combine the two RNAs together to make a single chimeric RNA. And they demonstrated that this chimeric RNA shown on the slide can work very well in vitro to cleave plasmid DNA in a test tube. So we thought if you can combine the two RNAs together to make a chimeric RNA, this can help us simplify the system. So we tested whether or not a chimeric RNA can work as well as when we reconstituted both of the CRISPR RNAs into a human cell. And so across the human EMX1 locus, we targeted 5 different regions, and we compared the efficacy. So on the right, you can see that although for one of the target, number 1, the chimeric RNA can cleave the human genome, but the other ones, the chimeric RNA didn't work, whereas the 2 RNA worked much more robustly. So that was very puzzling, and we compared the 2 RNA sequences again, and we realized that the chimeric RNA has been significantly truncated. And so we thought maybe some of the secondary structures that are on the tracer RNA are actually important for getting the system to work in the human cell. And so we then started to optimize the guide RNA, and we extended the 3' region on the chimeric RNA to include both of the hairpins. And so the first thing we found out using a northern blot is that the extension of the guide RNA significantly increased the abundance of the guide RNA within the human cell. And this also had a corresponding increase in the nuclease activity. So across the 3 different EMX1 targets shown on the bottom of the slide, we can see that inclusion of the first and both of the hairpin can significantly increase activity. And to some extent, it's even higher than using both guide RNAs at the same time. So this sums up the system. So you provide a Cas9 nuclease along with the guide RNA, and the guide RNA can be reprogrammed to target any genomic sequence as long as it's flanked by a small motif called the PAM sequence. In the case of the S-pyogenese Cas9, this PAM sequence is very short, it's NGG, and so on average you can find a targetable site almost every 12 base pairs within the human genome. So here's one application of the CRISPR Cas9 system. So using Cas9 and introducing a double-strand break within the EMX1 locus, we can at the same time provide a single-strand oligo. And the oligo will carry a small mutation, and through homologous recombination, this mutation can get incorporated very precisely into the EMX1 locus. And in HEK cells or human embryonic stem cells, we can achieve 27 or 6% of recombination respectively, and this is achieved without any form of drug selection or enrichment. So compared to without introducing a double-strand break, this increases efficiency by at least 4 or 5 orders of magnitude. So one important thing is to increase the specificity of the Cas9 nuclease. And so one of the methodologies that we developed is to convert Cas9 into a DNA nicking enzyme that only cleaves a single strand of DNA. And so very analogous to the way that talons or zinc finger nuclease work. So now we can require 2 different guide RNAs to target a single region. And this doubles the number of DNA bases that need to be specifically recognized by the Cas9 nuclease. So it's still possible that the Cas9 nicase and the single guide RNA can nick DNAs at off-target locations, but single-strand breaks are mostly repaired through the base excision repair pathway, which doesn't introduce an indel mutation. And so here is one example where we compare to see whether or not using double nicking can really increase the specificity. So by using 2 different guide RNAs, sgRNA9 and also sgRNA1, where sgRNA1 has 5 previously identified off-target sites, we first compare to see if single guide RNA and the wild-type Cas9 can achieve the same level of efficiency as if we use both guide RNAs and the Cas9 nicase. So on the left side, you can see a surveyor plot where you can see that on the very first lane, the double nicking Cas9 nicase along with both guide RNAs can achieve about 24% indel. And then on the right, you can see for lanes 4 and 5, the wild-type Cas9 with individual guide RNA can achieve 25 or 32% indel. So these are similar levels of indel efficiency at the target locus. And then if we compare the indel efficiency at the off-target location, then the story is quite different. With the wild-type Cas9 and the single guide RNA sgRNA1, you can detect indel mutations at all 5 off-target sites. But when you switch to the Cas9 nicase along with both guides, you no longer detect indel mutations. And so using this method, we can increase efficiency by up to 1,000-fold compared to just the traditional wild-type Cas9. Of course, this is a very rapidly moving field, and many other laboratories are also developing new ways to increase the Cas9 efficiency. So by combining Cas9 with any number of these efficiency improvement methodologies, we can get a fairly specific genome editing. So one application that's very powerful using the Cas9 system is that we can target many, many genes very easily. And so this allows us to build a genome-scale gene knockout library to study gene function across the genome. And so 2 of my postdocs, Ofer and Neville, developed a method by computationally designing guides to target every single gene within the human genome, and then synthesizing these guides using oligoarray DNA synthesis, and then cloning them to generate a viral library. So by applying this library of viruses onto the desirable cell line, you can generate a population of cells that has a mutation in every single gene within the human genome. Individually, of course, each cell would only carry a mutation in a single gene. And so by applying a selective pressure onto these cells, you can then start to identify what are the mutations that actually derive different kinds of biological phenomena within these cells. And so we apply this methodology to look for mutations or genes that would confer drug resistance in a subtype of melanoma. And so for this particular case of melanoma, where the tumors have a mutation in the BRAF gene, there is a drug called PLX4720 that can be quite effective. So on the bottom, you can see a patient who has this V600E BRAF mutation. The person can have tumors quite extensively, but by providing a PLX drug, the tumors can basically all dissolve away after 15 weeks of treatment. But unfortunately, the tumor cells are extremely vicious, and so they can develop mutations very rapidly. And within 24 weeks after treatment, the tumors will come back. And at that point, we no longer have a good way to target these tumors. And so we thought maybe we can use the CRISPR library to see what are these new mutations that the tumor cells acquired to make them resistant to the PLX drug. And so by taking cells that were infected with the CRISPR library, we applied the PLX drugs onto the library and then looked for the cells that were able to survive. And then by sequencing the guide RNAs within these surviving cells, we found specific genes or guide RNAs targeting specific genes were enriched in the surviving population. And these genes corresponded to NF2, MET12, COL3, NF1, and so forth. And so these are a list of candidate genes that may be potential drug targets for developing the next round of chemotherapeutic drugs. So compared to a methodology that many labs have been using based on shRNA, the CRISPR system seems to be more robust. So on the left side, if you look at the top 100 targets, the statistical significance value by the rigor method seems to show that there is an increased level of significance for each one of these candidate hits. And so if you look into the data a little more closely, you can find that for the top 10 candidates, for example, the multiple guide RNAs that were targeting the gene, they all consistently showed up. So most of the top candidates have 100% agreement among the guides, and then the other guide RNAs or the other genes had over 50% concordance across the guide RNAs. But the story is very different for shRNA, where you only have less than 50% concordance, and for even the top hit, less than half of the guide of the shRNAs agreed with the target. So this suggests that with CRISPR, you can get higher sensitivity and also likely higher consistency. So the fact that CRISPR can completely knock out a gene, whereas shRNA only provides partial knockdown, also confers functional differences too. So on the left side, you can see that using CRISPR, you can see a significant change in the growth phenotype of the cells under PLX, whereas on the right, with NF2, you don't see this growth phenotype difference. So what this means is that for genes where even a very small residual amount can provide us the full biological function, CRISPR has an advantage. It can fully deplete the protein, and then that's the only time when you can start to see the biological effect for those genes. So we're continuing to improve the library, and then most recently we reported in Nature Methods that you can use the CRISPR lentiviral vectors to increase the viral titer. And so using two forms, either a single vector or both vectors, or two vectors, one encoding Cas9 and another one encoding the guide RNA, you can now produce even higher titers, and that will be very useful for doing large-scale screening studies. And all of the libraries are now available through AGI. The new libraries also includes new guide RNAs to target microRNAs. So now you can identify or screen a different class of genetic elements within the genome. So finally, we're continuing to improve the Cas9 system. So one of the things we have been doing is to take a structure-based approach to increase specificity or activity of Cas9. So earlier this year, in collaboration with Osamu Noriki and Hiroshi Nishimatsu, we reported the crystal structure of Cas9 complex with the guide RNA and also a single strand of the target DNA. And so based on the structure, we're now making more structure-guided engineering of the nuclease to make it more efficient. So finally, just in summary, the CRISPR system provides a easy-to-use technology to both edit the genome, and also many labs have been developing ways to use CRISPR Cas9 to be able to modify the epigenetic states within cells. So we created an online website at genome-engineering.org, which describes reagents and protocols, and also there's an online discussion forum where researchers can pose questions and then interact with the community to get advice or feedback. And so here's just a fun picture of all the students in the lab working to prepare samples for distribution. So now, with the help of Agene, as well as some of our own distribution efforts, we're able to get this reagent out to several thousand labs around the world. So finally, I want to acknowledge our very significant collaborators, both Luciano Osamu, Phil Sharp's lab at MIT, Rudolph's lab at MIT, and then and the Genome Perturbation Platform at the Broad Institute, and also all of our funding agencies who made the work possible. So thank you very much for listening.
Well, thank you, Dr. Zhang, for that very informative talk. And we are now going to have a few minutes for Q&A. So we have several listeners who have submitted questions, and we'll go through them. If you do have any questions, you can type them in, and we will try to get to as many as we can. The first question we have is about how long the components of the Cas9 system stay active in the cells.
Yeah, thank you for the question. So the question is about what is the stability of the Cas9 components within the cell. Unfortunately, we don't have a very clear idea on the half-life of the protein within the human cell. But at least biochemically, the protein seems to be quite stable. And so in the cells, I think, based on our experience, it can last for at least a day or a little longer even.
Great. Okay, and now we have several questions we might be able to combine into one regarding off-target rates. So one question is if it's possible to estimate the off-target rate of the CRISPR-Cas9 system in ES cells. And a related question is regarding if there's any sense of predictability when it comes to off-target lesions using bioinformatics, or any ways to easily discover where the off-target indels actually are. So I will press control.
Yeah, off-target is a very important question. So first of all, the off-target effects seems to be higher and also is concentration dependent. So if you're using large amounts of DNA transfection and you're expressing the nuclease and the guide RNA for a long time within the cells, then the off-target, then you tend to see more off-target. So when you are doing a very critical targeting experiment, one of the things I would recommend is to titrate your amount of DNA transfection to a lower level. By using the lowest possible amount with a sufficient editing efficiency, you can minimize the off-target effect. In addition to that, there are a few different ways to increase the specificity. So one of them is by using the double nicking strategy that I described during the presentation. And then more recently, Keith Zhang's group at MGH also reported that you can truncate the guide RNA from 20 base pairs down to 18 or sometimes 17 to also increase the off-target, to decrease the off-target activity. And so what you could do is to combine both of the methodologies together using both double nicking as well as truncated guide RNAs. And together, the off-target activity can almost be undetectable. Whether or not you can predict off-target mutagenesis is also an important question. It seems like based on evaluating a large number of guides and also testing the effect of those guides on mutated targets, Cas9 will cleave DNA that shares similarity with maybe 2 or 3 mismatch. Will cleave off-target sites that have maybe 2 or 3 mismatches. And so what that means is computationally you can try to take the human genome sequence or any type of genome sequence and computation identify all the other sites that share high levels of similarity, say matching 18 or 17 or maybe sometimes 16 bases. And then what can be done is by doing targeted sequencing in the edited cell population to see are there mutations within those target sites. Cas9 doesn't target all those off-target sites, even though they share high levels of similarity. And right now, it's a little difficult to predict which one of these off-target, highly similar off-target sites actually have mutagenesis. So, in addition to that, there are also ways to be able to use Cas9 to see whether or not there are regions of genome that are bound by Cas9. So you can mutate Cas9 to become a catalytically inactive enzyme and then do chip analysis on the cell and then look at what are all the targets that are highly bound by Cas9. And then for those highly bound targets, you can also do targeted sequencing to see if there is any off-target mutation. But overall, I think the most important parameter is really not to introduce too much of the enzyme and really overexpress the enzyme. And then combining the careful controlling of the dosage along with improved double nicking or truncated guide RNA methodologies, you can really be fairly comfortable about the specificity of the enzyme.
Great. Okay, now some questions about the GeCKO library. One is, are there any plans to develop it in an array format? And the other is if there's any information about efficacy in C. elegans.
For the GeCKO library, so far what we have been developing are pooled libraries. And these pool libraries are convenient because you don't have to require very large infrastructures to be able to store them and to be able to carry out the high throughput screen. Right now, we haven't been developing array libraries, but there have been other groups, especially nonprofit or commercial groups, that are thinking about developing array libraries. So I think hopefully some form of array reagent will be available soon. In terms of efficacy in C. elegans, my lab has not directly worked on C. elegans, but there have been work published by multiple groups that show that you can achieve efficacious indel mutations within C. elegans. Homologous recombination so far doesn't seem to be extremely efficient, but I think it's still possible.
Okay, great. Well, speaking of homologous recombination, here are two questions about that. One is, do you test HDR in vitro prior to microinjection? And if so, is there a transfection method you suggest? And the other one is, if there are any methods to improve homologous recombination for gene repair.
In terms of homologous recombination and for embryo manipulation, we do test HR templates in vitro. And for doing that, we usually do nucleofection. And particularly when we are using single-strand oligos, we tend to use massive nucleofection to transfect the cells efficiently. The key for doing HR is that you need to get the templates directly into the nucleus. And so that's why nucleofection seems to work a little better than lipofection in our hands. Now, how do you increase the efficiency? One of the things we found is for small mutations, single-strand oligos tend to be more efficient than using double-strand DNA templates. And then for double-strand templates, if you can insert a drug selection marker, then by doing drug selection, enriching for the edited cells, the efficiency can be quite high, sometimes 50 or even 80% successful HR in the target cells.
Great. Here is a pretty short one. The question, first of all, says, thank you for sharing with the scientific community. And the researcher would like to know if you have any count of the publications resulting from other labs that use your CRISPR technology.
Yeah, that's an interesting question. Unfortunately, I haven't counted how many publications, but there definitely has been a lot of interest, and we've been trying our best to provide both online help and also getting the latest reagents out to everybody through Agene as soon as possible. Thank you for your question.
This question concerns recommendations for gRNA design for knockout and how you approach that. And they specifically ask if it's possible to load a 40 base pair sgRNA to target and cleave the ends of a particular gene. But I think the general question is about strategies for knockout.
For sgRNA design, there are a few things that are important to consider. One is, given that we know Cas9 can cleave at off-target sites, it is best to first computationally look at the guide sequence you have chosen and make sure it doesn't share high levels of homology with many sites across the genome. And so that's the first minimization step that I would do. And then second, for the guide RNA, it seems to be that for U6 transcription, you need to start a guide with a G. And G will ensure that U6 can transcribe the guide RNA at a high level so that you can get efficient editing within the cells. There have been a few very interesting papers, one from Jinsu Kim's lab, published in Nature Methods, that describe, depending on the specific sequence where you make the double-strand break, the indel that's generated may be predictable. And so, given that work, there may be ways to choose specific target sites to increase the rate of generating a frameshift mutation as opposed to a different kind of NHEJ product that doesn't lead to a gene knockout. So these are some of the considerations. And then also, based on some of our chip studies, it seems that having more AT-rich sequences toward the 3' region of the 20 base pair guide can also increase or reduce the number of off-target binding within the genome. Although some of that may be coupled with how efficiently the guide RNA can be transcribed within cells. So I guess just to summarize, the first thing I will do is just computationally try to minimize the number of homology sequences to the guide throughout the genome. And then number two, if you're using U6 transcription, make sure to start it with a G. And then also the same considerations for controlling concentration, and then maybe even trying truncated guide RNA can help increase the specificity.
Great. So the next question is asking for a comparison between CRISPR Cas9 versus TALENs and the TAL approach.
So comparing the Cas9 system with the TALEN and zinc finger systems, CRISPR is much easier to use. So to retarget the enzyme to a specific region in the genome, all you have to do is synthesize a short oligo to target that region. And so from an ease of use perspective, Cas9 is much more easy to use. From an efficiency perspective, so far it also seems that Cas9 is very much more efficacious. When we're generating knockout cell lines, oftentimes we find that Cas9 can generate homozygous, so mutating both alleles for a given gene. And I think the efficiency on average is higher than the average efficiency of TALENs or zinc finger nuclease. From a specificity perspective, using the optimizations that's possible, like double nicking or truncated guides, and also controlling the concentration of Cas9, I think we can achieve probably similar levels of specificity compared with zinc fingers and also TALENs. Although we still need to do a side-by-side comparison to really directly assess the specificity across these different kinds of nuclease platforms.
Thank you. Okay, we have time for just a few more questions. The next one is kind of a general question. Are you aware of any other RNA-guided DNA interactions in mammalian cells?
That's an interesting question. To my knowledge, I can't think of any off the top of my head, but there are just so many fascinating mechanisms in nature that I'm sure there are probably other RNA-guided DNA targeting features within not just the human cells, but probably many other organisms as well.
Thank you. The next question is regarding how much work has been done using CRISPR Cas9 technology in the case of plant systems.
There have been several examples of using Cas9 to edit different types of crop species, anywhere from tobacco to rice. And many of those studies have been published in Nature Biotechnology and also other journals. And so, so there has been a lot of applications of CRISPR in plants. Of course, editing plants opens up a lot of opportunities to generate both very important crop species as well as other useful plants with important biotechnological traits. So it's definitely a very exciting area with a lot of development.
Great. Okay, we're going to do two more questions. The first is asking about permanent integration of the Cas9 enzyme into a cell's genome, and does that affect later generations of cells?
It doesn't seem to be a very big problem. So we have generated using low titer or low MOI infection of cells, lentiviruses, so that cells can express Cas9, and the cell line seems to be able to propagate and be able to go through many passages. And so this is one thing that we do for doing genome-scale screens. By providing a cell line that already has Cas9, we can just generate a library of viruses to express guide RNAs, and that can work quite well. We have also generated a knock-in mouse where a single copy of Cas9 is integrated in the genome and is under constitutive expression. And that mouse has crossed and bred for multiple generations and also seems to be healthy, reproductive, and there are no abnormal things that we can observe in any of the tissues.
Great. Okay, well, we're going to do one more question now. There are a few of you we did not get to your questions. We will provide them to Dr. Zhang, and we'll try to follow up at a later time. The last question is regarding the double nicking strategy compared to wild-type Cas9 with respect to homologous recombination, and is one better than the other?
In terms of comparing the homology-derived repair efficiency of double nicking and wild-type Cas9, I think both can be fairly efficient. There are some cases where we do see maybe even slightly more efficient HR using double nicking, although we haven't done a very thorough test across many different targets to see if that trend generalizes. But I think you can certainly achieve HR quite efficiently using double nicking.
Okay, well, once again, I'd like to thank Dr. Zhang for his presentation and answering questions this morning. And that concludes this morning's seminar. Thank you for attending.